Hello, welcome to Hide Science Lab Series Episode 9. If I'm looking a lot more HD, it's because I got a fancy new camera we're going to try out today for the first time. So I'm holding this lab up, or this paper up, because this is our lab today. This is chromatography. This is paper chromatography. So what we are doing here is we are separating the ink by the properties of the dyes within the ink. So I'll explain a little bit more about that once we get going, because we do have like a little time delay we'll have to get through. So bear with me, I'm going to get this camera situated and then we'll roll from there. Alright, so here we are back, and this is kind of our lab guide here. As you can see, we got some images of what's going on and kind of what our results will be, how it works. This gives us some background information to our chromatography. How do we use it? Why are we using it? What solvents? What are the properties of the solvents? Because how their molecular structure is really affects the results of what happens. And then on the back here, this is our guide the students get. We've got a procedure, our data, how to solve for the RF factor, and our post-lab questions. So basically what we do is we are going to take 4 chromatography papers, and we are going to use them, and we're going to test one in alcohol and one in water. So we're going to kind of get that rolling real quick. So we're going to use one Sharpie marker. This is a permanent ink. We're going to go up about 1 to 1.5 centimeters on these, about the same spot. And we're going to kind of fold this to sit down in our jar. We're going to put one in the alcohol beaker, and then we're going to do the same thing here, and one in the water. And this is just going to fold this so it hangs on there. So then we're going to pick kind of any other Crayola color that we want. We will do the same technique up about an 1.5 centimeters, and we will make a line. We're going to hang this as well, one in each. Okay, so we're about to pour our water and our alcohol into the jars. What is very important is that we do not want the water level or the alcohol level to get near this line, because that will cause the ink to diffuse out into the liquid. So we want to make sure that the liquid level is just here at the bottom of the paper. So we'll do our alcohol into our alcohol cup. Yeah. And then we will roll our water into this one. Alright, so now we're going to have a little bit of that time delay that I spoke about earlier. So the water is being absorbed by the chromatography paper up through the paper. And then this is being absorbed, the alcohol is being absorbed at the same time. So we're just going to have to wait for that process to occur. And as we do, just to talk a little bit about it. So this is our alcohol beaker. This is our water beaker, this one. So water is polar because we have one side of the molecule that is positive, the other side of the molecule that is negative. So this gives us the polar effect, like the Earth with the north and south pole, or a magnet with a positive and negative pole. So we have charges on the opposite side. Alcohol has this kind of slight polar leg down here, but overall the molecule is not polar, it is non-polar. So the different dyes used in the ink have different properties of polarity as well. And the more similar they are to the water or to the alcohol, then the more attracted the dye will be to those liquids and will separate them. And in biotechnology, so we are a manufacturing career for the most part. So there is always usually some designed to make a product. So this is a very basic tool for analyzing the properties of chemicals, and also for maybe purifying or separating different things based on their properties. So we will get more advanced with this as we go, but this is kind of where we are starting here. So as the liquids are absorbed, we will get different results. So this has not really gone long enough, but just to kind of give you an idea, the paper on this one is wet up to this point. You can kind of see the water line. So this Sharpie marker is not separating much in the water. We're getting a little separation there. And here we can see this marker is separating. We're starting to get some yellow, some light green. So we're seeing the colors separate here. If we were to continue to allow this to go, it would continue to separate. In our alcohol, this is a slower process. It's not, don't have quite the properties of water, so it's not moving as quick, but we're seeing some separation here, and then we're seeing better separation with our Sharpie. So just kind of based off of that, we can see that the ink in this was our alcohol results, this was our water. The ink in the Sharpie is more similar to the alcohol than it is the water, because the alcohol it's attracted to the alcohol and it is moving it. And then with the water and the alcohol here, these color inks are more like the water, so they're more polar dyes. The final steps of this would be to calculate the RF factor, which is highlighted here, which will do the distance traveled by the ink divided by the distance traveled by the solvent. So we measure those out on our chromatography strips using a ruler. We'll measure from our origination point to how far the inks moved and how far the paper got wet. So we'll finish that. We'll get our calculations done. And that is it for our ink chromatography background lab. We'll see y'all next time with the lab series number 10, which will be another type of chromatography.